All right, good afternoon, everyone. So thanks for joining for the session Neuroscience Frontiers, Exploring Cognitive Dynamics and Brain Connectivity. So we've got some really cool talks for you today. Please, I'll just remind you to submit questions if you have them via the app. So the first speaker we have today is Ashley Tyrer from Aarhus University, who will be talking to us about towards interoception as a transdiagnostic biomarker of health-harming disorders.
Hello. So I'd like to start by saying that I am not Micah Allen. He was supposed to be giving the talk today. He's my supervisor, but unfortunately he can't be here. So unfortunately for you guys, you've got me instead. I'm Ashley. Thank you for the introduction. I'm a postdoc in Micah's lab, the Embodied Computation Group. And what we mainly focus on as a lab is the study of interoception. So, how do I change the slide? There we go. So what is interoception? So at the most general level, interoception can be described as the physiological sense of the homeostatic processes and internal condition of the body. And we can sort of divide interoception into two modes, if you will. So we have implicit or pre-conscious interoception, and we also have explicit or metacognitive interoception. So in implicit interoception, one might perceive an external stimulus differently if that is experienced during a cardiac contraction versus a cardiac relaxation. And this might lead us to encode that stimulus differently. So this isn't something that we're always conscious of at this implicit level of synchrony between the brain and the body. Explicit interoception, on the other hand, is essentially when we can actively perceive or become metacognitively aware of those visceral sensory and motor signals. So for example, when you start to feel your tummy rumbling, when you're getting hungry, or another example is when you can feel your heart start to race when you stand up in front of a group of people like this and give a talk. I'm feeling very interoceptive right now. So all of these signals sort of give us an indication of the bodily activity on a daily basis. So how does our lab investigate this? We can start by trying to improve measurement strategies. So for example, the development of new psychophysical tools or computational models to aid in the examination of interoceptive processes. And then we have discovery science. So the large-scale neuroimaging data collection that will enable the validation and exploration of these processes. And then, of course, we have intervention. So pharmacological intervention, lifestyle changes, deep brain stimulation, all of these things will aid in the targeting and improvement of any health-harming disorders that might arise from aberrant interoceptive processes. So back in 2020 and 2021, mainly during the COVID-19 lockdown, when apparently the people of Denmark had nothing better to do than take part in a massive neuroimaging study, we ran a neuroimaging study. And it was over 500 people, and we collected data from a large number of modalities. As you can see on the screen, this is a very complex figure. So I'm just going to highlight two of the studies that was born from this mammoth data collection project, and then also I'm going to finish off with some of the work that I've done in the past year or so. So I mentioned explicit and implicit interoception. So I'll start with an example of explicit interoception. Namely the fingerprints of respiratory interoception in the microstructure of the breathing brain. So our lovely postdoc Nia collected this work. So she ran a direct investigation into how the microstructure of the breathing brain might be influenced by respiratory interoception. And she did this by the multiparametric mapping analysis, or MPMs, of quantitative MRI. So this measures indices such as cortical myelination, myeloarchitecture, and iron concentration. And she did this by also combining it with our Bayesian psychophysical approach, the respiratory resistance sensitivity task, which measures aspects such as interoceptive precision, interoceptive accuracy, metacognition, and breathing capability. So what did she find? Interestingly, in the postcentral gyrus, she found a significant increase in correlation in myelination with slope, which is essentially interoceptive precision. So on the left here, we have the psychometric functions of all subjects with the mean in yellow. And the slope is basically the gradient of this psychometric function, which is an index for interoceptive precision. Similarly, in the anterior insula, we found the converse, so a negative correlation with the level of myelination in this region to the interoceptive threshold, which is basically the midpoint of this psychometric function. and the threshold can be used as an index of interoceptive accuracy. And this is obviously very interesting because obviously this is not functional imaging, but a future functional imaging study might be able to reveal some of the neural correlates that are actually involved in this association. And now for something completely different. So that was a good example of how we've investigated explicit interoception, and now we're going to switch it around and look at how we've investigated implicit interoception, so namely the stomach-brain coupling and how that is associated with mental health. So, during the VMP, or the Visceral Mind Project, we also recorded electrogastrography, or EGG, which basically measures the contractions and relaxation of the stomach. And this was recorded in combination with 14 minutes of resting state fMRI. So from this, we calculated the stomach-brain phase coupling, and then this was parcellated across 209 regions using the TFUMO atlas. On the other side of things, we also collected 37 different psychiatric symptom inventories, so things like depression, anxiety, stress, quality of life, and well-being. And then Leah ran something called a canonical correlation analysis, or CCA. And what CCA essentially does is finds a maximally correlated pattern across these two sets of multidimensional variables. It sounds very complicated, but basically what happens is you take these two variables, and then using a 5-fold cross-validation method, you calculate a set of weights, and then these weights are used to look at the correlation between these two variables. So, very fancy method, but in principle, a very simple result. We found that higher stomach-brain coupling is significantly correlated with worse mental health. So higher scores of depression, higher scores of anxiety, higher scores of stress. And then also the converse, that lower stomach-brain coupling loadings were found to be associated with better mental health. So higher scores in well-being and higher scores in quality of life. And here we're just showing the top 5 TFUMO regions with the absolute highest correlations in those stomach-brain loadings. Okay, so to further summarize the findings, we also averaged our stomach-brain loadings across the Yo7 network. And what we found was the networks that were most highly significant were those in the attention and control networks. And we also averaged the psychiatric scores across mental health categories and found the maximal correlation between the highest stomach-brain loading regions, so the attention network and the control network, were most maximally correlated with high depression scores, stress, and anxiety. So, of course, we have to put the disclaimer that this is a correlation analysis, not causation. We're not making any statements about causation here, but it could pose some very interesting future work on finding out what exactly is the directionality of this. You hear a lot about people who suffer from anxiety, also suffering from stomach issues and digestion issues. So I think it's very important to consider the rest of the body in combination with those mental health disorders, and it plays a large role in the ongoing feedback loop between brain and body. And finally, another different third thing. So this is a bit of my work. So in the past year, I collected data from a large MEG study looking at predictive processing under central versus peripheral noradrenergic blockade. So to do this, we used 2 different beta blockers. We used propranolol, which is a centrally acting beta blocker, which means it can cross the blood-brain barrier. And we also used bisoprolol, which is peripherally acting, meaning it cannot cross the blood-brain barrier. But it has very high specificity for the heart and also the lungs. So we used a face affect prediction task. I'm not going to use the acronym for that one, but basically for this task, there's a cue, and with a certain probability has an association with either a happy face or an angry face. And the participants basically have to learn that probability association. And there are, of course, reversals in that, so then they have to kind of deconstruct their model of the task and rebuild it to relearn the task structure post-reversal. So here's some very preliminary results. I'm going to do a bit of baiting here, but basically this is kind of all the results I'm going to show in this presentation. The kind of behavioral accuracy effect, we can see that there is a significant effect of the high precision prediction error, and that has a significant effect on the accuracy of subsequent trials. And we only see that under the propranolol condition. For placebo and bisoprolol, it's pretty much the same. To be continued. So I have a poster. It's number 927. If you wanted to just kind of discuss some of the more preliminary analyses and go into some of the neuroimaging analyses, then I will be at the poster tomorrow, so please come and visit. So just to summarize, the brain cannot be considered an isolated entity. I know I've presented 3 sort of mini projects, all very different with very different findings, but the main take home from each of these studies is that we can't treat the brain like a lonely island. It's kind of part of the grand archipelago of the human body. We have to consider this ongoing feedback loop between the brain and the body and back again. This leads to discussions about embodied phenomenology. So the example I used about people with anxiety also suffering from stomach issues. Traditionally, these things have kind of been treated as side effects. I think future research would highly benefit from considering these things as part of the feedback loop that modulates these health-harming disorders. And so by understanding brain-body synchrony in more detail, we might further our understanding of the body's role in mental health. There. So I would like to thank you all, the lab, and of course our glorious leader, Mike Allen, and also our collaborators, Ignacio. Ignacio is here at OHBM. I believe he gave a workshop on Sunday, but he's absolutely fantastic. So if you have any questions about the stomach research, please find Ignacio. And I'd also like to thank everybody who was involved in the mammoth data collection, because it was a lot of hard work. If you have any technical questions about any of the studies, I've provided the email addresses for each person who led those studies, and also their poster number. Those posters will be there tomorrow, I believe. And I just had to include this comic, because whenever I go to a conference, I'm always reminded of it. So yeah, with that, thank you very much.
All right, we've got time for one quick question. There's nothing on the app, so I might take the chair's privilege. I'm really curious what you think the predictions might be if you investigated people with a vagotomy that had been performed, say, for some form of illness. Do you think that that would affect your ability to interocept? Or do you think you'd find other ways around that signal?
That's a tricky one. It depends on the context of the vagotomy. So if it's something that's developmental, then compensatory mechanisms may have been developed throughout life. We see that in a lot of other disorders. But if that's something that's happened after that kind of critical period of development, if it's happened in adulthood, then we might not see those compensatory mechanisms, and we might just see a dropout of that function. So it really depends. Thank you.
All right, join me in thanking Ashley. Thank you.